Again, today we have a topic of synapses, and our topic is transmit of impulses across synapses. And again, here we have lesson plan. We will introduct synapses and its structure, mechanism of transmit the impulses through the synapses, and we have chemical and electrical synapses. And here, this is one typical chemical synapse. And synapse is connection between two neurons. It's a junction between two neurons, and usually the synapse is formed by the axon of one neuron ending, and the axon ending will be become bigger, larger, and here this one is the presynaptic neuron. Axon ending, axon terminal, and here the presynaptic axon terminal has a definite intact membrane. It's called presynaptic membrane. And here in the axon terminal, we usually have two important structures. One is mitochondria, and next one is synaptic vesicle. And mitochondria is to synthesize energy, and here it helps in the synthesis of neurotransmitters. And the synaptic vesicle is to store neurotransmitters. Again, here we have synaptic cleft between presynaptic membrane and the postsynaptic membrane. We have a junction, we call it synaptic cleft, and after that we have a postsynaptic membrane. It includes postsynaptic neuron, and the membrane of the postsynaptic neuron is called postsynaptic membrane. And it can, you can see here it contains some receptor proteins, and receptors, we have different types of receptors, and here we mainly introduce two receptors. One is cholinergic receptors, which combined or binds with the acetylcholine, and we have adrenergic receptors, which bind with adrenaline and noradrenaline. So again we can subdivide it into two types. Cholinergic receptors, we can divide it into muscarinic and nicotinic, and adrenergic we can divide it into alpha and beta adrenergic receptors. It depends on the neurotransmitters and the location of organs. In the chemical synapse, there is no continuity between two neurons because of the presence of space, which is called synaptic cleft. Here it is, and the action potential reaching the presynaptic terminal and the cause release of neurotransmitter substance from the vesicles of the terminal, and neurotransmitters reach to the postsynaptic neuron through the synaptic cleft, and cause development of potential change at the postsynaptic neuron. And in here we have one scheme. Here this is normal, kind of non-excited, and here is excited. We call it excitatory postsynaptic potential. And you see here we have lots of acetylcholine is released into the synaptic cleft, and some of them will be destroyed and some of them will be reused by the synaptic terminal, and we will talk about it later after 32 slides. And here we'll come back to neurotransmitters, and here we give some example of neurotransmitters. Acetylcholine, for example, noradrenaline, and we have GABA, glycine, histamine, and we have serotonin, we have dopamine. It's a chemical substance which is synthesized in the presynaptic neuron axon terminal, and it acts as a mediator for transmission of nerve impulses from one neuron to another neuron across synapse. And again, here we mainly introduced acetylcholine. Before that, I want to explain some information about neurotransmitters. And neurotransmitters, it must be found in neuron and produced in neuron. And again, it because it's found in neuron, produced in neuron, it must be released by neuron. And again, after releasing it to the synaptic cleft, it must be bind or act with target area, for example, postsynaptic membrane, to generate to develop to produce some biological effect, such as action potential, postsynaptic excitatory potential. And after that, it must be inactivated. Choline again, and we said it transmits impulses from the nerve fiber to the CNS, and again transmit impulses from the preganglionic sympathetic nerve to the ganglion, from parasympathetic nerve to the effector, from motor neuron to effector neurons. And one word, acetylcholine is a transmitter substance or at the neuromuscular junction and the synapses. It's excitatory, and again, it's released by most of the following nerve endings. Number one is preganglionic parasympathetic nerve endings, and again postganglionic, yes, also pre and postganglionic parasympathetic nerve endings, and again it's released by the preganglionic sympathetic nerve fibers, and some of postganglionic sympathetic cholinergic nerve fibers. And again, this kind of nerve fiber will supply sweat glands, and some skeletal muscles, vasodilator skeletal muscles, and again, brain can release acetylcholine. And again, noradrenaline from the impulses from the postganglionic adrenergic sympathetic nerve fibers to the effector organs. And here we have some kind of enzymes which can destroy neurotransmitters after it acts on the postsynaptic membrane. And here we have cholinesterase. It will destroy acetylcholine in the synaptic cleft. And here we have a monoamine oxidase. It will destroy the noradrenaline. And again, we have here we said neurotransmitters are classified into two types. We have excitatory neurotransmitters, and we have inhibitory neurotransmitters. Excitatory neurotransmitter is a chemical substance which is responsible for the conduction of impulses from the presynaptic neuron to the postsynaptic neuron. And inhibitory neurotransmitters is again, it's a chemical substance which inhibits the conduction of impulses from presynaptic neuron to postsynaptic neuron. And here we have some examples of excitatory neurotransmitters, and again, some examples of inhibitory neurotransmitters. Here we have a classification of synapses, and by location we have axonic synapses and two axon of two neurons synapses, and we have axodendric, one axon of one neuron synapse with dendrite of next neuron. And again, we have dendrodendritic, we have axosomatic. Soma means body. One axon of one neuron is synapse with body of next neuron. And we have neuromuscular synapses or neuromuscular junction, and the axon of one neuron is synapse with muscle tissue. And by effect or by function, We have excitatory and inhibitory synapses. Excitatory synapse, it will transmit impulses from presynaptic neuron to the postsynaptic neuron, and inhibitory synapse, it will be stop or inhibit the impulses from the presynaptic neuron to the postsynaptic neuron. And again, we have bimode of impulse transmission. We have electric synapses, we have chemical synapse, and we have a mixed synapse. And again, one mechanism which we can develop EPSP, it means excitatory postsynaptic potential, or to let the impulses transmitted from presynaptic neuron to postsynaptic neuron. We will go through all this kind of steps. Let's talk about it one by one. And the first one, arrival of action potential in axon terminal, and after that presynaptic membrane will be depolarized. And here the mechanism is because it is depolarized because of influx of calcium ions from the ECF into the axon terminal, and the releasing of excitatory neurotransmitters such as acetylcholine by means of exocytosis. And again, after it released, it will be released into the synaptic cleft. And again, after it, it will be passage of neurotransmitters through the synaptic cleft. And again, these neurotransmitters will bind with corresponding receptor proteins present in the postsynaptic membrane to form neurotransmitter receptor complex. After that, we will have developed the non-propagated EPSP. This is local potential, it's not real action potential. Then we have many neurotransmitters coming, and it will combine with lots of receptor proteins. It will generate real action potential. This is how an impulses is transmitted from presynaptic neuron to the postsynaptic neuron. And again, here is a scheme. Yeah, you can see how it is transmitted. And again, we said we have excitatory and inhibitory synapses. This is how it is inhibited, And again, the same action potential is arrived at the axon terminal. Again, it will be depolarized, same as in excitatory synapse, but here it will release inhibitory neurotransmitters, but here it released excitatory neurotransmitters. So for example, GABA. And again, it will permeate the hyperpolarization at the postsynaptic membrane because of it opened ligand-gated potassium channels, and potassium is efflux of potassium into the extracellular fluid. And it's because of, sometimes it's because of opening of chloride channels and influx of chloride into the neurons. Then the inside of postsynaptic neuron is more negative, and it will become hyperpolarized, and it is difficult to generate an action potential. Then we have IPSP, inhibitory postsynaptic potential is development, and the synaptic transmission is inhibit at this site. And here we have some properties between properties of synapses, and we have compared chemical and electrical synapses. And again, in the chemical synapses, impulses are transmitted by the release of chemical neurotransmitters, and in here it's transmitted by the flow of current ions. And here chemical synapses, it can only conduct one-way conductions. The impulses can only be conducted from the presynaptic neuron to the postsynaptic neuron. And again, here it's two-way conduction because there is no presynaptic neuron and terminals and the postsynaptic neurons and the receptors. And again, chemical synapses liability is very low. and this is electric synapse variability is high. And here chemical synapse, we have synaptic delay because we need to synthesize and transmit it into the synaptic cleft, and here it's absent. And here chemical synapse, we have summation. And again here no summation, is only excitatory. And here I said I translated it by open way. It's kind of up to one impulses is transmitted through the synaptic cleft or through the synapse. The next impulses is easier to transmit it, through this synaptic cleft. And again we have fatigue. At chemical synapses. Here no fatigue, and the depletion of it's because of the depletion of neurotransmitters. And we have selective chemical synapses, selective sensitivity to pharmacological and some biological substance. And this is how we use some pharmacological substance to control our nervous system. And its sensitivity to temperature change, and again it has presence of local depolarization. And here IPSP and EPSP, it's local depolarization because it's non-propagated. And again, that's all. Thank you for listening.